Hi guys, a very warm welcome to all of you in Gurmantra Academy. I'm your educator Saranya, and today it's going to be your plant and animal biotechnology class. It's going to be a continuation. So we're going to see a lot of topics today, and you are also going to have a quiz. So we'll see some topics in the beginning. We'll continue with quiz topics. Okay? So be ready guys, okay? Alright, so we'll learn about cell line today. Okay, what is a cell line? When a primary culture, what is the meaning of primary culture? It is something which you obtain from an animal directly, like an explant, okay? Directly from an animal, you take the cells and culture it, that's primary culture. You keep on passaging it, okay, again and again and again. Keep on subculturing it or passaging, okay, then it becomes like a cell line. It becomes a secondary culture or a cell line or a subclone, okay. So what is the meaning of finite cell lines? Finite cell line means they have a limit up to which they can do cell division. They have a limited lifespan after which they die. Okay, after which they die, they lose their ability to proliferate. First, they will lose their ability to proliferate because there is some genetic modification which has taken place, and they will not divide anymore. They'll be alive for some time, they will not divide, and that stage is called as senescence, and after that they'll die. Okay, after that they'll die. These are called as finite cell lines, and they are primarily derived from primary cultures. Okay, continuous cell line. Continuous cell lines are ones that has undergone transformation. What does that mean? It means that they have become so much modified, that is there have been so many mutations, genetic modifications, they have been completely transformed into a different type of cell. It's not like they are a complete different cell, but the way they divide, the way they are having mutations is all very different. So what does continuous cell line mean? It means that they are transformed cell. They can divide continuously, infinitely, which means there's no limit at all. You cannot stop them. They can divide for a very long period of time till they gain senescence. So such transformation is because of mutation. It can occur either spontaneously, or we can cause these kind of mutations using virus. Okay, virus can cause a cell to become a cancer. You would have heard about virus induced cancer, right? In which the cells will be dividing indefinitely. So cell lines can also be created in such a way. Okay, and they are immortal cell lines. They do not die. They just go on and on and on. So these type of cell lines are different from normal cell lines in that they are less adherent, which means they will not attach itself to the substratum. They grow very fast, and even if the nutrition is little bit less, they'll grow to higher density. Okay, and they will show different phenotypes, which means their characteristic feature will be different than the original tissue from which they have been derived. And mostly these type of continuous cell lines will be grown in liquid medium suspension culture. Also they have a tendency to form multi layers, like for example, this is your culture vessel. It'll form like this is a mono layer. Okay, this is a mono layer of cells, but continuous cell line has the ability to form multiple layers. They do not understand contact inhibition, which means when two cells are in contact, they signal with each other, they contact with each other, saying that okay, there are not enough space, also the nutrition is less, let's stop dividing for a while. This is what is called as contact inhibition. This property is not present in any of the cancer cell. So does continuous cell lines have this property? They do not undergo contact inhibition. Some of the common cell lines in which usually research take place is like MCF7, which is a breast cancer cell line, HL60 leukemia, HeLa cells, very famous, you guys should have heard about HeLa, human cervical cancer cell. It was this name was derived from the patient from whose cervix they remove these cancer cells. So you guys have a short homework. Find out what is the full form of HeLa. This is the name of a person. Okay. Next you have primate cell line. Primates means like monkey. Vero. Vero is the kidney. It is the kidney cell line. So why do we use cell line? What is the application? There are several applications for cell lines. The first one is vaccine production. Vaccine production requires the cells to grow indefinitely to a larger scale. If you're going to use primary cell line or a secondary cell line or a finite cell line for that matter, after certain passages, they'll undergo senescence and eventually they'll die. But if you're going to use a continuous cell line, then it's going to be great use. The cells will grow indefinitely, you'll get large amount of produce and thereby you can make large quantity of vaccines. Okay, for example, now for the coronavirus, they use adenovirus. Okay, the cell line will be used. Okay, so this is a very common technique. Next is virus cultivation and study. Okay, so virus, you know about that, it's an intracellular obligate virus. Okay, so what does that mean? It can grow only when it has the host. So in order to study virus, we have to culture them. So the culturing of virus is more convenient, easy and economic when you use cell culture instead of actually injecting the virus into the animal. That is little bit difficult to even do some research. But if you're going to grow the virus via the cell culture, it will be very, very easy. So viruses are usually grown through the cell culture technique. Next, cell and molecular biology. So any kind of research you have, it starts with your model organism. So model organism, you will start from cells. Okay, you'll start from cells. You'll study its physiology, biochemistry of the cell, like what are the metabolic processes going on, about the aging, about different organelles, toxicity, mutagenesis and carcinogenesis. So one way is that it will tell whether, for example, if you're talking about a drug, whether it will be useful for a person to treat a particular disease. So first they will start with cells. They will check if the drug is able to penetrate into the cell and cause the effect. They'll check for toxicity, they'll check for the concentration, they'll check for mutagenesis, carcinogenesis. So any kind of research, drug discovery, everything starts with your cells. In cancer research, see, normal cells can be transformed into cancer cells by using several methods. I've told you, like virus, you can use some chemical mutagens and they become cancer. So they are more or like the cancer which exists in a patient's body, cancer patient's body. So now you can do research on that cells, like what are the target drugs you can use to cure the disease. Gene therapy, a very important function and application of cell line. You can replace the non-functional gene by functional gene using cell therapy. Okay, gene therapy, cell therapy can be done. Immunological studies. So immune cells, cytokines, lymphoid cells, all of these things has interaction with the antigens, the disease causing agent. We can study them well using cell culture technique. Okay, so next we are going to understand about is transgenic animal. So transgenic animal are genetically engineered animal. Their genome has been modified, altered in such a way that they will have a trait which we want. Our gene of interest will be inserted into the genome of the animal, and it will be able to produce any product we want it to produce. That is the technology we talk about when we are saying transgenic animals. So the first transgenic animal was the sheep, Dolly. She was created in Scotland in 1997 by the nuclear transfer technique, which means the nucleus of the donor cell was injected into the recipient cell egg, that is egg cell. Okay, so mammary cell, they took mammary cell and injected its nucleus into the egg cell. Okay, okay, that is what they've done. So cytoplasm of the egg cell and the nucleus of the mammary cell was fused. Now this cell was implanted into a surrogate mother, and then it eventually developed into Dolly, a lamb. She was a transgenic lamb because she was containing a human gene. Okay, another homework for you. What is the human gene that uh, she was containing? Okay, that is the Polly and not Dolly. Polly, what is the human gene that Polly was containing? So this development of Dolly, basically development of Dolly is called as cloning. They understood cloning, and development of Polly is transgenic. So from these events, developments, they understood that yes, they can create cloned animals, and they can also make these animals carry human proteins. So what they did was they removed the udder cell from the sheep, and its nucleus was taken, and a unfertilized egg, okay, unfertilized egg was taken, and DNA from that was removed, that is nucleus was removed. So cytoplasm of egg cell and the nucleus of the udder cell was fused, and then it became a like a zygote, a complete cell which is good enough to cause a clone. Then it was implanted into a surrogate sheep, and she gave rise to a clone sheep, and that is Dolly. So normal physiology and development. In transgenic animals, what happens is a foreign gene is introduced into them. So there is kind of a growth factor that is altered. So these animals helps in the study, like gene regulation study and their effect in day-to-day function. We can understand diseases. A specific gene can be inserted into these animals that will cause a disease, and they can act like a model animals. For example, mice will be used as a model for Alzheimer or a cancer or a heart disease, myocardial infarction. So these are the diseases which can be induced into the mice, and we can study treatment in them. Next is biological products. We get lot of products, like for example, the milk that contains the human gene alpha lactalbumin. It can be actually coming from a cow. A natural cow can produce this. Okay. Next is vaccination. Vaccination is one of the most important thing that you can obtain from a transgenic animal. Okay, so the next topic we are going to understand about is microcarrier and macrocarrier. So they are nothing but they support the growth of the adherent cells. Like it is also a cell culture technique, but they are going to they are like a carrier. Okay, they are a carrier on which the cells will grow, especially adherent cells, which means they'll stick onto the surface, act like a substratum and grow. So microcarrier size is like 125 to 250 nanometer. It will be like a sphere. So what are the materials which can be used? Dextrin, glass, polystyrene, acrylamide, collagen, alginate. These are some of the materials that can be used. Okay, so a microcarrier is a support matrix that will allow the growth of the cells which are adherent to these microcarriers in a bioreactor, where you can grow large amount of cells. Similarly, microcarriers are regularly used to grow protein producing or virus generating adherent cells. So in this way, you can either produce biologics, which means the proteins which has therapeutic value, or you can produce vaccines. So a liquid-based assembly method was developed by the Shen et al. For assembling cell-seeded microcarriers into diverse structure. Neurons were seeded in these microcarriers, and they actually develop the neural network like 3D shape, and this is called as potential tissue engineering technique. So advantage is that the scale up is very easy. Large quantity of cells you can grow. You can control the growth. Okay, you can do that. Incubation time is less, and there is not much of a labor. Labor work is not involved. So there are two types of microcarrier culture. One is that density of the microcarrier is greater than the culture media itself. So the microcarrier density is greater than the culture media. That means the growth of the cells is sensitive to the microcarrier concentration. So agitation and oxygen is required. If the density of the microcarrier is less than the culture media, there is no limitation of growth for the adherent cells. So usually second type is used. Second type of microcarrier culture is used. Okay, macrocarrier. This is also similar to microcarrier only. Only thing is its size is very big. It also supports a matrix. Okay, so oxygenation is good, nutrient exchange is good, and the bioproduct yield is also pretty high. So can you see here? Can you see here? This is the matrix. Okay, this yellow color straw-like structures are the matrix, and these are the cells. Can you see here? These are the cells which are attached to the matrix and they are growing. Microcarrier, macrocarrier. So nutrients can dissolve in these microcarriers and macrocarriers, and the cells will take up, and any waste material they are releasing can go out of it. Very simple and effective. So the next topic we are going to learn about is cryopreservation. So this is a method where we keep the cells, tissues and biological samples at a sub-zero temperature, that is minus 196 degrees Celsius. We store them, we preserve them for a very long period of time. So what happens is at such low temperature, the biological activity of the cell is reduced, it stops and usually dies, okay, at minus 196 degrees Celsius. That is why cryopreservation helps the cells to survive freezing and thawing. Okay, so the cells formation, see, basically when you decrease the temperature, normally without any cryopreservation, you're decreasing the temperature of the cell, which is like at 37 degrees Celsius, eventually to minus 196 degrees Celsius. What will happen? Ice crystals will be formed. Why? Because the cell's content is 95% water. Water, when temperature goes down, it will form ice. Ice will form like a crystal and it will break the cell membrane and the cell will die. Okay, so we have to carefully regulate the freezing rate, and we have to choose a freezing medium in which there is no ice crystal formation. Only then the cell can survive at such sub-zero degree Celsius. Okay, so cryopreservation means cold preservation. You can preserve a sample for up to 20 years. The temperature is minus 196 degrees Celsius. MCQ question. MCQ question. Next, what can you store? You can store embryo, oocytes, spermatozoa, ovarian tissues, any kind of cells you want, you can store. Advantage is that very good preservation for long period of time, and there won't be any kind of contamination or disease. So cryoprotectants, which means they are some kind of a liquid medium which is used to protect the cells from forming ice crystals and dying. Okay, so in liquid nitrogen medium, glycerol, DMSO, PEG are some of the cryoprotectants which can be used. What these does is it can easily penetrate inside the cell and decreases its freezing point. Okay, it will decrease the freezing point to level that they will be mixing with water. Okay, mixing with water which is present in the cytoplasm, decrease the freezing point. When the freezing point of a particular liquid is decreased, then even though you lower the temperature, they will not freeze. So these kind of substance have, these cryoprotectants have that property. So what is the procedure? The procedure is that you will collect the sample in a sterile container. You will mix the cryoprotectant like DMSO, dimethyl sulfoxide, or glycerol like 10 to 20%. Then you will start freezing. Like how do you freeze? Like 1.5 degrees Celsius you will reduce per minute. Okay, till from 20 degrees Celsius to minus 80 degrees Celsius, you will reduce 1.5 degrees reduced per minute. Okay, then will be rapid freezing from minus 80 degrees Celsius to minus 196 degrees Celsius. You will do rapid freezing, which means 6 degrees Celsius will be reduced per minute. Then when it comes to minus 196 degrees Celsius, you can store it. Cryopreservation steps. So you will get the material like your biological sample. You will add the cryoprotectant. Then you will freeze them. I told you the method of freezing in liquid nitrogen container, and when it is needed for use, you will have to thaw them. Thaw them means you have to give warmth. You have to give the warmth from minus 196 degrees Celsius to, you have to bring it to the normal temperature. So you have to give the warmth, and you have to control the rate of cooling in such a way that again crystallization does not happen. So what is the benefits of cryopreservation? You can actually store the sperm and egg. So for fertility treatments, in vitro fertilization and all, they can use this. Labor and all is not required much because it's a very simple procedure. Genetic contamination will not happen, and the integrity of and the value of the material will be there for a very long period of time. The germ plasm of the endangered species can be stored so that we can revive them back and bring those species which are going to be extinct. And biological samples can be preserved for a very long period of time without any contamination, and it will prevent the genetic drift. Okay, we have studied about genetic drift, so it can prevent that. So application, so seed bank, you can store seed. Gene bank, you can store the gene. Blood transfusion, in vitro fertilization, organ transplantation, artificial insemination in the animal husbandry. Then storage of the rare germplasm like extinct animals or endangered species. Freezing of the cell culture, very important. Very frequently it is used in the labs. Then conservation of endangered species of plant and animal. Then biodiversity conservation. Mostly for the conservation purpose, cryopreservation is used. Okay, so this is your homework question. Next, we are going to understand what is a stem cell. So a stem cell is a cell with a very unique ability that it can develop any kind of cell in your body. They can replace the damaged tissues, and you'll have a new tissues in that place. So that is what a stem cell does. When it divides, it gives rise to a differentiated cell and a copy of stem cell. So you will get one differentiated cell. It can be a blood cell, a neuron, a adipocyte, a chondrocyte, and the other cell will be a stem cell. This is the self-renewal property. So when a stem cell divide, it will give rise to one stem cell, the same cell itself, and one differentiated cell. Okay, this is how stem cell, they basically divide. Pretty cool now. So okay, our body is made up of many different types of cells. Most cells are specialized to perform particular functions, like red blood cells carry oxygen around our body. They are not able to divide. They cannot divide because they don't have nucleus. Why? See, it's a very logic thing that a RBC's main purpose is to carry oxygen. Okay, it can carry a lot of oxygen. If nucleus is there, nucleus is a very big organelle in a cell, then RBCs, the weight of RBCs will become more, then its travel will not be easy. That is why when the RBCs become mature, they lose its nucleus, and their only purpose is to carry oxygen around in your body. So stem cells provide new cells for the body as it grows, and it will replace the specialized cells that are damaged. Okay, so they can divide again and again and again, making new stem cells, and when they divide, they give one type, that is a specialized or differentiated cell. Okay, so these are some of the very important MCQ questions we should know. MCQs, all of it is MCQ. Totipotent, it can give rise to a whole organism itself. So the cells from early 1 to 3 days. See, these questions they ask you in interviews also. Okay, like what are totipotent cells? So for how many days in an embryo the cell will be totipotent? Please know this. Okay, these are the questions they definitely ask you in interviews. I've been asked these questions myself. That is, I'm telling you. Okay, 1 to 3 days, very important. Okay, next is pluripotent. Pluripotent means they can give rise to more than 200 types of cells, more than 200 types of cells. Okay, but not all cell types. So they will develop after 5 days till 14 days. Yeah, you can simply remember 5 to 15 days. Multipotent. They can give rise to quite a number of tissues, not like pluripotent, but a little bit. Like for example, you have fetal cord blood and adult stem cells given rise to mesenchymal stem cells. Okay, so different types of stem cells. You have embryonic stem cells, the embryo itself, adult stem cells, and the induced pluripotent stem cells. Embryonic stem cells means it will come from an embryo which is 5 to 6 days old. Okay, they have the ability to form virtually any type of cell in the human body. They can form any type of cell. Okay, embryonic germ cells means they will produce egg and sperm. Adult stem cells means they are undifferentiated cells, they are in our body surrounding, but whenever there is a cell damage, these stem cells will differentiate and repair the damage. But their reproduction ability and differentiation ability is very limited. So embryonic stem cells are pluripotent in nature. They can make any type of cell in your body, but they cannot make the whole organism, because embryonic stem cells, they do not include the placenta. Okay, trophoblast. Okay, that is not actually required too much of developmental biology, but yeah, you just know that they cannot make a whole organism, but they can make most of the cells. Next, when it comes to adult stem cells, they are multipotent, like bone, neuron, blood, cord blood. These type of cells it can form. Next is specialized cells. These are the specialized cells. So induced pluripotent stem cells are nothing but they have been induced, artificially created in the lab, reprogramming the stem cells. Okay, just like embryonic stem cells, they are also pluripotent in nature. They can develop any type of cell but not the whole organism in itself. Next is stem cell therapy. Why are stem cell therapy important? If there is any kind of damage in your body which cannot be cured, or it is like a permanent damage by disease, injury, or some kind of genetic condition, stem cells has the ability to create new good cells which can be transported to the diseased region and you can replace the damaged parts. Okay, so these are some of the conditions, like examples I've given you here. Blood stem cells are used to provide a source of healthy blood cells for people with some kind of blood condition like thalassemia, cancer patients. Okay, skin stem cells can generate a good skin tissue which can be used to treat burns, acid attack patients, and the severe burns patient. Those type of people, we can treat them with skin stem cells. Next is muscle degeneration. If a retinal pigment epithelium is not working, you can use these kind of cells. This is one such example. Retina, this cell of the retina is impaired, so stem cells are differentiated into retinal cells. They are implanted into the eye. Now the retina is working fine. Okay, okay, 1 second. Oh god. Okay, so what I was trying to say is that your plant and animal biology, biotechnology topic is over. I've completed all the topics. Now we're going to have a quiz session. Okay, we are going to start a quiz. So get ready, have a notebook, pen. You can just mark your answers. Okay, let's start a quiz session. You'll have 1 minute for each question, and after that you will get the answer. I'll show you the answer, don't worry. The production of secondary metabolites requires the use of. Okay, so the correct answer for this question is option D, cell suspension. Next question. For callus differentiation, what are the hormones required? These questions you know, right? We have studied these in plant biotech, in cell culture, plant cell culture. Okay, so the correct answer for this question is auxin and cytokinin. Next question. What is DMSO? Just now we studied. You don't even have to think a lot. We just studied it now. Okay, the correct answer is option B, cryoprotectant. Next question. Totipotency refers to. The correct answer is option B. Actually, what I would say is, I would not even say option B is correct. I would say a totipotent cell can give rise to a whole organism itself, not just an organ. Okay, next question. In tissue culture of parenchyma, mitosis is accelerated in the presence of. So, which of the following hormone is involved in mitosis, cell division? Correct answer is auxin and cytokinin. Next question. Haploid plants can be obtained from. You can basically obtain this from uh, the germ cell culture. So here the germ cell is anther. Anther is the correct answer. Next question. Which of the following vectors is used in crop improvement and crop management? So the correct answer for this question is agrobacterium. Next question. Which of the following growth hormones produces apical dominance? Apex. Easy questions, guys. So you don't even need 60 seconds. Think quickly. Okay, for correct answer, you guys can give yourselves 2 marks. Okay. And if your answers are wrong, then minus 1 mark. Total it. Okay, whatever is your score, total it, and you can post it in the group, the Telegram group. Correct answer is option C, auxin. Next question. Hybrids. They are talking about hybrids. Hybrids are produced by the fusion. Okay, 30 seconds is actually enough for this question. The correct answer is option A. Next question. Which of the following mediums is composed of chemically defined compounds? Very easy question. Very, very easy question. Chemically defined compounds will be present in your... Okay, I'll give you 30 seconds. 1 minute is too much, guys. For these are very simple questions. I'll give you 30 seconds. Okay, so the correct answer for this question is option C, synthetic media. Next question. Which of the following chemicals are most widely used for protoplast fusion? फ्यूजन के लिए कौन से केमिकल्स यूज किए जाते हैं? अगेन आई गिव यू 30 सेकेंड्स एंड आई रिवील द आंसर फॉर यू. इसमें सोचने वाली उतनी बड़ी बात नहीं है, डायरेक्ट आंसर्स हैं, डायरेक्ट क्वेश्चंस हैं. Okay, so the correct answer for this question is PEG, polyethylene glycol. Next question. What is a callus? Do you need even 30 seconds for this question? See, guys, don't think that I'm asking you these questions. It means it will come in gate or it has already come in gate. These questions are to check whether you've understood the concept, whether you are listening in the class. It is just a practice session based on whatever I have taught you now. Okay, so we will prepare you for even difficult question, okay, in the test series, in the PYQs. Okay, so you have a lot of opportunities, so don't think in that way. So the correct answer for this question is option B, undifferentiated cells, dividing cells. Next question. Which of the following is best suited method for the production of virus-free plant? For the production of virus-free plant, meristematic culture is used. Okay, next question. Batch cultures are type of suspension culture where? I'll give you 30 seconds. It's very easy. Batch. In the name itself, its work is defined. नाम में ही काम है. 5 more seconds. Okay, so the correct answer for this question is option B. Next question. Hairy root culture for secondary metabolite production are induced by transforming the plant cells with what? This is also very easy. It is a very good gate question though. Okay, so the correct answer for this question is the rhizogenes, Agrobacterium rhizogenes. Okay, that is it guys. Your test is over. I hope you guys would have enjoyed the quiz and the lecture. Plant biotechnology is over by now. We'll move on to the next topic in the next class. Okay, so if you have any queries, you can always contact us. Thank you so much guys. Have a nice day. Keep preparing well.